Assalamu alaikum wa rahmatullahi wa barakatuh. Dear student, this is Dr. Zaim Al-Jabri, and today's lecture is about sterilization and disinfection. In today's lecture, we are going to learn about the different mechanisms and techniques that we use to achieve the rate of zero infection. But actually, this time in history when we talk about sterilization and different practices of antisepsis and disinfection, we really appreciate the importance of it when we are talking about time where there is a threat, global threat of COVID-19, and you see everywhere those social distancing notes and also this emphasis on the hand hygiene. So after this lecture, I hope you also can have a general idea about what are the different important chemicals or detergents that we use to achieve sterilization in the hospital environment. So in this lecture, we are going to define different terms with the relation to sterilization, disinfection, and antisepsis. We're going to also discuss the various physical and chemical methods in order to achieve bacterial control and prevention. So, as I said, there is this time in history where we need a lot of sterilization to happen. But imagine what happens before. So you can take a look at this picture on the left-hand side. This is an operation room before. So this was in 80s, 70s. See the doctors, how they are actually surrounding the patients. They are wearing their everyday clothes. There is no antiseptic whatsoever. And they are only using carbolic acid spray as antiseptic at that time. And you can see the difference now on the right hand side where you have these big theaters with full sterilization, cleaning, antisepsis and disinfection. Doctors are going through a very long process of cleaning their hands, washing, and also draping of the patient. After that, there is a proper cleaning of the OT or operation theater, and this actually takes place at real time or at the same time. So you can see here that through time we appreciate what is happening here because we are reducing the number of bacterial or the hospital acquired infections. And there also previous times, there were times where a lot of ladies used to die after giving birth, and then they found out that the surgeons were giving birth to these ladies at night. They were actually dissecting cadavers or dead bodies, and because they did not recognize at that time that these are ways of transmission of a disease, a lot of ladies used to die and also their babies used to die. This is not happening anymore. Luckily and thankfully, we have this all sterilization techniques. Okay, so why do we need this sterilization and disinfection? Usually in every semester when I ask this to the students, they are very good at pinpointing the purpose, but sadly we cannot really interact face to face. I find this very difficult also for me. But you all know that we need to prevent transmission of disease through microbes from one patient to another. That's why we need to know all the details of sterilization and disinfection. And that's why we have what we call standard precautions. And in the standard precautions, we have, first of all, hand hygiene, hand hygiene, hand hygiene. And I always emphasize on this step. It's very, very important. And in my practicals, I usually demonstrate to students the steps of hand hygiene. I make sure that they wash their hands before and after coming to the lab. Second one is respiratory hygiene while coughing, and you know the coughing etiquette when you have to cover your hands, your mouth with your elbows while coughing. And there is safe injection practices, and you know here as nurses that you never recap a needle because this will put you at risk of needle prick and then hurting yourself, transmitting any contamination from the needles through the blood of the patients to yourself. A fourth important standard precaution is by proper disposal of needles and scalpels, and because you will be working in hospitals, there will be proper disposal using specific places where you put the sharp and scalpels, and there are specific other waste management. And if you remember in the previous slides of the practical, we learned that we have three types of general waste. Please go through them, remember them very well, because you are going to practice them when you go to the hospital. So, what are the important hand washing steps? I used to ask the students, and I used to ask one of them to come in front of the class to demonstrate this to me. And they are very good at this. I think you, as nurses, um, you have to understand that these washing steps are, it has to become like the alphabets to you. When you say, when you come to the hospital, you have to do this and perform them at minimum of 40 seconds. And ideally, it should be up to 60 seconds, and you know that you should do them very well. So I'm not going to go through them because I'm assuming that you know them. But I'm going to ask you. So now let's go through the most important definitions in this lecture. The first one is sterilization. What do we mean by sterilization? So sterilization basically means that we want to remove all microorganisms, including bacteria like bacterial spores. And as you remember from the last lectures, that bacterial spores are highly resistant to killing by the heat or nutrition deficiency. But in sterilization, where you boil, not boil, but you actually autoclave the different equipments, you actually can kill the spores. Example for this is medical equipment sterilization, as you see in the picture. In disinfection, however, you don't remove or kill all microorganisms, but you are trying to kill almost all of them, but this is not possible. You might kill most of them or many of them, but not all of them. And sometimes also you just inhibit the growth, not kill them. And this is also because the bacteria that have spores can actually survive the disinfection. It will not be killed. A good example of this is when you want to draw some blood from your patient, what you do is you bring a 70% ethanol, or we call them alcohol swab, and you try to clean the skin of the patient, and then you put your needle in. So this is one example where you disinfect the skin, but you don't sterilize the skin. I hope this is very clear to you. Now let us move to the definitions, other definitions. So because we said disinfection, so we need things to disinfect. We call them disinfectants. So these are chemicals or antimicrobial agents that we apply to non-living objects. to destroy the microorganism. What do you mean by non-living object? It basically means that you put it on your table, you clean it, your chair, your surfaces in the hospital. While antisepsis is actually antimicrobial agent or substance that you apply on a living tissue like the skin of your patient to make it antisepsis. And each one of them will have different agents, and we will discuss them in this lecture in more details. So, if we want to roughly divide the methods for sterilization and disinfection, they actually are divided into two major categories: chemical agents and physical agents. What do we mean by chemical agents? There are chemicals, and the chemicals are the agents that have different mode of action. So when you see MOA, it basically means mode of action, similar to the pharmacology drugs. So there are agents that might disrupt or disturb or break the cell membrane, like alcohol that we use on the skin of the patient, like phenols, detergents that we use to clean the floors, and also some of these chemical agents can modify proteins, which means that they degrade or break the proteins on the cell membrane of bacteria and viruses, like chlorine. You know chlorine very well. It's the bleach that we also use at home to make, we apply on the white clothes to, to remove any pigments or stains. And also we have iodine that can modify protein. I know that you know iodine very well. Heavy metals like sulfur nitrates are example, silver nitrates, sorry. And also we have hydrogen peroxide. All of these are examples of agents that modify protein, and also formaldehyde and glutaraldehyde. These have similar mechanism of action. Also, we have gas sterilization. So the previous ones are in sprays or liquids, while gas is actually found in a form of a gas. And a good example of gas is ethylene oxide and formalin gas. Physical agents means that we don't use chemicals, we use heat, for example, radiation. And radiation, they are divided into either non-ionizing radiation like ultraviolet light, ionizing radiations like X-rays and gamma rays. Also, we have another example of physical agent, which is filtration. So in this lecture, we're going to go through all of these. So now we move to the first chemical agent, which is alcohol. As we mentioned earlier, it causes disruption or break of lipid-containing cells. As we learned that the cell membrane of bacteria has a phospholipid bilayer. So it gets broken by the alcohol, and also it can cause protein denaturation. A good example of this is the 70% ethanol or isopropyl alcohol that we use in the hospital. Why 70%? Because actually this is the maximum activity. We cannot use 100% because it's more powerful. No, actually we dilute it in water to make it more powerful, because we need these water molecules to make the alcohol penetrate the tissue of the bacteria very easily and be able to kill it more effectively. So alcohol can also kill bacteria and fungi, but usually does not kill spores. And we use it as antiseptics, as I mentioned earlier, to clean the skin before we do blood collection or venipuncture. So let's discuss detergents. So detergents is basically means the cleaning agents that we use even at home, and also at the hospital. And these detergents are composed of two parts: the lipid, which is the hydrophobic portion, and there's the polar hydrophilic portion. If you remember from chemistry basics, that hydrophobic means it likes lipids, but it does not like water. So hydrophobic, so it's afraid of water. Polar hydrophilic means that it likes water. So one part, like in the picture here, you can see that the hydrophilic portion, actually the non-hydrophilic, non-polar portion will go to the lipid component of the cell, and then it causes it to emulsify, similar to what the soap does, and it makes it degradable, or it can disturb it and then break it. Good example of this is benzalkonium chloride. It is very commonly used in the hospital, and it's a part or an example of quaternary ammonium compounds. And we use it on the skin as antiseptic, and also can use it mixed with Lysol, which is like a floor detergent, to disinfect the floors in the hospitals. If you can see here in the picture, these are the common detergents that you see at home. But you can see also from the picture here, we have a comparison between two plates where you grow bacteria from the toilet that has not been cleaned. You see a lot of whitish stuff there or yellowish, that's bacteria. And after cleaning with Lysol, you can see almost all bacteria disappeared. So this is very effective at cleaning. Now let us move to the third type, which is phenols. Again, phenols are very commonly used in the hospital. They act by disruption of lipid-containing cell membrane and protein denaturation. So this is very similar to alcohol. And it's very caustic. What do you mean by very caustic? It means that it has the ability to destroy the skin. So you don't actually use it in itself. Usually what we do is we mix phenols with another agent like chlorine, for example, and you make what we call chlorhexidine. And if you go back to your, when you go to the hospital and you look at those pink soaps on the sink, you read the label, you find this word chlorine written. I'll show you a picture in a minute. So this is one example of these phenols we can use as hand scrubs at the labs and before surgery, and we use them for wound cleansing. This is also another type of chlorhexidine where we use it in the form of soap for bathing the patient and also for washing towels. This is the bottle that I was talking about. I'm sure you're familiar with this pink bottle. It has chlorhexidine in it. And I'm sure also you're familiar with Dettol, with all the forms of Dettol we use at home. It's again a chlorinated phenol that is very commonly used at home and also in the hospital. Now let us move to another example of this detergent, which is chlorine. You can see from the picture, it's Clorox, so it's the same thing. You can have it in liquid form, or in the lab we have it in the tablet form. So we can just put it inside water and it will dissolve immediately. How does this chlorine work is that they act as oxidizing agent. So what basically this means that it needs a protein, it targets the protein structure of the bacteria and oxidize it. And after oxidation, it will be destroyed. As I said, example is Clorox or bleach. It's the hypochlorite. And this kills bacteria, viruses, and fungi. But again, like alcohol, it does not kill the spores. So for spores, you need something more strong. This actually is used for disinfecting these surfaces. Sometimes also we can use this for purifying water supply. If you realize that if you have this tap water at home and sometimes you smell the chlor on it, it's actually because it's the government's add chlor to the water to kill any pathogens and also viruses and fungi. Also, it's used to treat swimming pools. So when you go to the public swimming pools, you notice there is a very strong smell of chlorine. And that's very common, again, as a detergent to wash out or to kill any pathogenic microorganisms. You have to remember that this chloride is a corrosive agent, which means that it can also again harms the skin, so you have to be very careful not to touch it. The ones that we use in the water is very diluted. That's why we don't get harmed. Now let us move to the other chemical agent that we commonly use in the hospital, and it's very clear from the picture. It's iodine. So what is iodine? Again, it's similar to the phenols. It's an oxidizing agent. Sorry, it's similar to the chlorine, oxidizing agent, and it has two forms. The first one is tincture of iodine, which is 2%. It's a yellow stain, and it can cause skin irritation, so it's very, very strong. You have to be very careful. And then we have the povidone iodine, which is less irritant. And I think you are very familiar with the uses from the picture here. You can see that it's used as a skin antiseptic. So when you apply it on the top of the skin before collecting blood culture. So there's a difference between venipuncture and blood culture. Venipuncture is what you do when you try to collect blood from your patient in a normal, for a CBC count, usual blood work. But for blood culture, you need more sterilization, you need more cleaning for the skin. So you need something stronger. You can use alcohol swabs, but on top of that, you can also use povidone iodine. And also you use this iodine to clean the clean the genitals before inserting the intravenous catheter, because you, again, as I said before, you need to make it very sterile to prevent your patient from reintroducing any bacteria by inserting the catheter. And again, the picture shows you here that surgeons, before going to surgery, they apply this on their skin. They wash it very well, like you can see here. There's this scrub brushes that has a lot of iodine in it. They wash it like soap, and then they also apply it on the top of the skin of the patient, and then they drape it. So apply it, and then they clean it, and then on top of that they apply the draping, which is the dressing, before they start operating on the patient. Now we move on to formaldehyde or glyceraldehyde. A lot of chemicals today, but I'm sure you will remember them because they are very important to learn. So formaldehyde is a toxic substance because it actually denatures or breaks protein and also DNA. And it's actually very commonly used in pathology, where they dissect tissue and freeze them and keep them, stay for years. They preserve these tissues in formaldehyde. And formalin is basically 37% formaldehyde in water. Glutaraldehyde is something like formaldehyde, but it's 10 times more effective and less toxic. So we use this, which is basically will cause less harm to us. How do we use it? We use it because it's very strong, so we sterilize the respiratory equipments. We use it also for sterilization of endoscopes and hemodialysis equipments. So you can see from the picture the endoscope tubes before inserting inside the the oral cavity and the digestive tract. You need to make it very sterile. Endoscopes of different types, and also hemodialysis means that these are the units that we use for kidney dialysis for the patients. So you make sure that they are very clean before you use them for any patient. Okay, now let us move to the last chemical agent, which is gas sterilization. So gas sterilization is ethylene oxide, and ethylene oxide is used for sterilization of any material that is heat sensitive. So anything that is made of plastic and metal surfaces and glasses, they can be very easily sterilized with this gas. And surgical instruments and plastics also, because if you put them under heat, you are going to destroy them. But here you can use only the gas, which does not destroy any plastic. And this is not liquids, of course, not for liquids, because it will be toxic for the liquids. This gas sterilization is very highly inflammable, so it means that it will immediately get burned if they are exposed to any burn. And they are irritant and carcinogenic, so they have the ability to cause cancer. So that's why we have this label that it's cancer hazard and reproductive harm. And only authorized personnel can use this equipment. Now we move on to the physical agents. So what are the most important physical agents? Here we are going to discuss the heat first as the first example. So what do we mean by heat, and how do we use it for sterilization of equipment? So heat is a form of energy. So this energy is what kills the microorganisms. And how does it kill it? By causing protein denaturation. And what basically this means is that it breaks down any protein structures inside the microorganism. So the bacteria cannot actually live it if the protein is destroyed. And also it can cause membrane damage to the microorganisms and also enzymatic cleavage of any DNA. So this reassures that most microorganisms can die. There are basically two types of heat. We have first the moist heat and dry heat. The moist heat means it's wet. So that's what you do when you boil or autoclave. So autoclave is the machine that you see in the picture here where you put inside any liquids and then you close it and then you leave it there for some time. There is also dry heat, so it's the same, but actually here we don't have water boiling. It's just the mechanical heat that is dry. I mean the mechanical dry heat that is produced out in the air of the oven. And there's flaming. This is what we use in the lab every day. That when we work on the bench, we actually use this type of a Bunsen burner. And fourth is pasteurization. So I'm not sure whether you are familiar with Louis Pasteur, who invented this technique. And we use pasteurization for sterilization of liquid like milk, for example. Okay, now we go through them one by one. So what do we mean by moist heat? It means it's wet. So the heat is actually wet because there is water that is heated and then it produces moist. This is similar to what we have in the kitchen when you have that pressure cook that I showed you in the previous slide. So the water here helps breaking hydrogen bonds of the proteins in the bacteria, and this is done by a machine called autoclave or the process called autoclaving. How do we make sure that this procedure is effective in killing microorganisms? Is actually we validate by using a bacteria containing spore or a biological indicator like a bacteria has spore Clostridium spores. So if the bacteria is killed and also the spores are killed, we are 100% sure that our technique is correct and we have achieved correct sterilization or autoclave procedure. That's why we use it to actually sterilize surgical instruments in the hospital, culture media, linens, which means that the sheets that we use for the patients, plasticware, solutions in water, biohazardous waste. This is the autoclavable disposable bags that we use in the hospital and also in our labs. It's plastic bag that we put any waste inside and then we tie it and then we send it for autoclave. This is what the machine looks like from the inside. So you basically put whatever you want to autoclave, let's say the bottle inside, and you have water that is circulating in the tubes. It's not in direct contact with the actually in the bottle, but actually it produces steam, and then the steam is going to exceed certain temperature, which is usually 121 degrees Celsius for at least 20 minutes or 15 minutes, and this produces a pressure of about 15 pounds per square inch, and this is actually similar to a pressure cook. You don't allow the steam to escape so that it will kill anything inside. In the kitchen, we use it to cook the meats and also different dishes, but it's actually the same concept. Here we kill bacteria, viruses, and spores as well. So this is the first technique that can kill all of these microorganisms, including spores. So what do you mean by the dry heat? So dry heat is similar to the autoclaves, but the difference here is that we only have hot air. We don't have moist water. And also the other difference is the temperature here is 180 for 2 hours. The previous one it takes shorter time. And this is operated by having electricity with a fan to circulate the hot air And we validate this technique by what we call a biological indicator. So we need to have Bacillus subtilis spores. So again, it needs to be validated. We make sure that the procedure is correct. So this is what the hot oven looks like, or hot air oven. Similar to this is the flaming technique that we use in the lab, where we have this wire loop. We expose it to the heat, and then we use it to culture the bacteria. How do we use this technique? We actually use it for glassware. The dry heat, we use it for glassware, anything made of glass. You can sterilize it by this oven. You can also use it to sterilize surgical instruments, anything that the surgeon can use, like scalpels and forceps, and also for chemicals like paraffins and glycerol. Now, there's another form of a physical agent apart from moist heat and dry heat, and flaming. The fourth one is pasteurization. So, what do we mean by pasteurization? As you can see from the picture here, there's milk that you are taking out from the animals and cows, and then you expose it to heating section and then cooling section very rapidly. So that's why what we use for sterilization of milk. The initial heating takes up to 62 degrees for 30 minutes, followed by rapid cooling. So you put it inside and then you cool it immediately. And this will actually what we also call rapid cooling. But there's another technique, which is the flash heating, which is only for 15 seconds. You are exposing the milk to 72 degrees Celsius. Why do we sterilize the milk? Because actually milk, if you drink raw milk, you are going to cause yourself an infection. Most commonly, it can cause Mycobacterium bovis, Salmonella, Streptococcus, Listeria, Brucella, and we see a lot of cases in the hospital, especially from Salalah or during Kharif season, when people go there and drink a lot of raw milk from the camels. They come with Brucella, and we will learn about the Brucella infection later on in this course. So this is what the machine looks like and how it's actually used for sterilization of milk. Okay, so another form of a physical agent for sterilization is radiation. What I mean by radiation is radiation energy. It has enough energy to remove an electron from a molecule, then it's ionizing and it has more energy. If it's non-ionizing, then it produces less energy. And in this picture, you can see the difference between ionizing and non-ionizing. So for example, ultraviolet light here that we are concerned about produces more energy, and it's ionizing because it can remove a molecule. In the next slide, we are going to take more examples. So this is what you see in the hospital sometimes. We have this ultraviolet light, and this is a form of radiation that we use to sterilize, or a sterilization technique. So how does this work? UV light actually works by acting on the DNA of the bacteria or the pathogen, and it causes damage to the DNA. And it has, if you are using UV light, it has what we call low penetration power. So because of this low penetration power, it does not penetrate effectively through these surfaces. So we only use it to sterilize the surfaces. So it will prevent the bacteria from growing because there will be no DNA replication. And we use it for cleaning operation theaters or rooms, the wards, the surfaces when they are not in use. So this basically means that if at night or if the operation theater is empty, you can switch these lights off, and then you apply the UV light on, and this will cause damage to any bacteria or pathogens that is remaining on the surface. But you have to be very careful again, because this is very damaging to the skin and cornea, so you don't really expose yourself to this light. Another type of radiation that is used for sterilization is X-rays, the same X-ray that we use for the patients for, so taking an image of the chest, for example. But this time we use it also for sterilization. Why do we use X-ray? Because they have higher penetration powers and they can penetrate the tissue and also the equipment, and has higher energy to break any DNA inside the equipment. Both X-rays and UV lights are effective, but they cannot kill bacterial spores. Only the bacterial spores can be killed by autoclaving or the dry heat. Why? Because you see here it has very low water content. The spores are very dry, and X-rays depend on and UV light also depends on the microorganism having a lot of water in it. How do we use it? Actually, we use it for heat-sensitive material like sutures, surgical gloves, and syringes, as the ones also shown in the picture. That's why also we call it cold sterilization, because there's no heat in this technique. The last technique that I want to talk about today is filtration. And what we mean by filtration, as you can see from the picture here, we have a syringe filter attached to a filter, and also there's these millipore filters. It's the white layer on the bottom. That is the filter or filter paper. So filtration is used for any heat-sensitive compounds. This means that if there is any liquid that you want to make it sterile, but you don't want it to be damaged by the heat, you can do filtration. So what it does is that it traps any particles that cannot escape the pore size of the filter. This includes, for example, sterilizing any IV fluids, vaccines, and antibiotics. We don't use them immediately on the patients. We actually receive them filterized. And this because if there is any bacteria that is dead, the endotoxin in the cell wall sometimes can still be effective in causing fever in the patient. So if you don't filter that, it might cause fever. So it has to be filtered before use. Of course, you are not going to filter it yourself because you have to receive it from the company ready filtered. And an example of the filter, like I showed you in the picture, is what we call nitrocellulose filter paper, which has different nano size, which is 0.22 micrometer. And this filter can trap all the bacteria and their spores and also endotoxins without causing any infection. So this is the end of today's lecture. If you have any questions, please let me know. You can send them by email, or you can also put your questions in the band group. Thank you very much for listening.